The transformation of big data into clinical knowledge isn't an instant process, but rather a complex journey that we can call the genomic data lifecycle. The example of ultra-rapid diagnosis, achieved by Professor Ewan Ashley's team, is currently an exception that applies to patients in intensive care who need quick genetic diagnoses to determine their treatment. In these cases, drastically shortening the genomic data life cycle to just a few days offers significant advantages over traditional tests, which usually take weeks. This journey, which goes from the lab bench to the patient's bedside, is called bench to the bedside and is orchestrated by bioinformatics and computational biology, which serve as the essential bridge between data generation and its interpretation. This life cycle is possible thanks to a complex ecosystem in which every stage affects the quality and final value of the data. Now let's look at the crucial stages of the genomic data life cycle. The first stage involves collecting the biological sample, such as blood or tissue. Proper preservation of the biological sample is vitally important. And this takes place inside biobanks, which are far from being simple warehouses. Instead, they are clinical infrastructures that ensure the quality, integrity, and traceability of the sample. Since the quality of the initial sample absolutely determines all subsequent analyses, biobanks are one of the pillars of precision medicine. And they ensure the availability of biological material for both current and future research. Now let's move on to the second phase, which is the generation of raw data. In this phase, the samples are processed using next-generation sequencing platforms, also known as NGS, used to generate an enormous amount of raw data. Just like in the first phase, this is a critical stage where non-biological variability can be introduced, which can significantly affect the analyses. Here, too, it's important to use standardized, controlled, and fully traceable procedures. The third phase is quality control and pre-analysis. It starts with quality control of the raw data obtained. This step aims to verify that the experiment was successful as expected, and it's a fundamental step to ensure the reliability of the information. At this stage, the first filters are applied to remove noise and artifacts from the process. The fourth phase consists of analysis and interpretation, where computational biology comes into play. During the analysis, programs and pipelines are used to extract meaning from the sequences, identifying, for example, genetic variants that could explain a tumor's resistance to a specific drug. Usually, this is the part that takes the most time. The last phase, which is no less important, is archiving and sharing. Both the raw data and the results of the analyses are stored in specialized databases. And the availability of these public databases is crucial for scientific collaboration and for validating results. As central points of reference, we have the U.S. ecosystem, the National Center for Biotechnology Information, and the European counterpart, Elixir, where various databases are interconnected to facilitate research. For example, you can start from the SRA, Sequence Read Archive, at NCBI, which is the largest archive of sequencing raw data, to trace back to a gene expression dataset of interest in GEO, Gene Expression Omnibus. You can do the same by visiting the ENA, European Nucleotide Archive, a European service that hosts sequencing data and connects with the SRA. Other NCBI databases, like DBVAR, archive structural variants of the genome, while PubMed contains information related to scientific literature. This integrated system makes it possible to track data from its raw origin all the way to its functional and clinical interpretation. Resources like NCBI, Ensemble, and UCSC also provide visualization tools, known as genome browsers, which allow you to explore these complex data sets in a spatial and functional context, making interpretation easier so you can draw conclusions. Data sharing does, however, have its own challenges. Resistance to data sharing and the lack of standardization represent significant challenges. This is not just a technical issue, but an ethical, legal, and cultural challenge that acts as a causal bottleneck, in fact, preventing the full realization of the value of big data. The data lifecycle is not a linear process, but an interconnected ecosystem where quality at every stage, from storage in a biobank to the final analysis, determines the value that can be extracted from it. Each of us owns our own genome and must protect it like a vigilant guardian, but only through the anonymous sharing of thousands of genetic codes we have the ability to decipher and understand the code of the individual. In conclusion, bioinformatics is not just a tool, but the conceptual pillar that supports the entire framework of precision medicine, acting as a bridge that connects data generation to value extraction. In fact, in recent years, data generation is no longer an obstacle, and the largest portion of the data cycle is devoted to analysis. This is something Professor Ashley's team knew very well. So, going back to the example of ultra-rapid diagnosis to win the race against time, the research team took advantage of nanopore technology, which is a third-generation sequencing method capable of producing long reads in real time. To speed up data analysis, which is the slowest phase of the process, a method was developed to send sequence packets to cloud analysis systems that use parallelization across many CPUs, or rather GPUs, drastically shortening analysis times.